Hello everyone, I'm Rochelle and today I will be presenting the second chapter of my PhD project entitled The Effect of Organic Amendments on the Severity of Apple Replant Disease in Subsequent Apple Orchards Replantings. Apple replant disease is the soil-borne disease that causes stunting of young apple trees when they are planted into orchards where apples or related crop species were cultivated previously. ARD is caused by a complex of microbes and nematodes. The long-term buildup of the ARD causative agents in the orchards during the lifespan of the previously planted trees causes stunting of newly planted young trees, which leads to decreased apple production. Currently in South Africa, ARD can only be managed in replanted orchards using fumigation, which is unsustainable and costly. A possible preventative strategy is the use of organic amendments for the suppression of the pathogens. Compost and mulches can potentially be used to reduce the buildup of the ARD pathogens in apple orchard soils and ultimately reduce the ARD severity in subsequent replanted orchards. The aim of this project is to determine if long-term organic amendment application can reduce ARD in new replanted orchards. The objectives are to identify trial sites with sufficient ARD, determining whether long-term organic amended soils can suppress ARD under glasshouse conditions, determining whether apple orchard tree roots growing in organic amended soil contain less ARD causative agents, and investigating the associations between soil health parameters and the ARD suppressiveness of organic amended soils. 6 farms were selected as potential trial sites for the pre-screening of the soil. All 7 blocks selected received 5 years of mulch, compost and mulch, or no amendments. We wanted to identify the presence of 3 of the most important ARD causative agents, namely Pratylenchus, Phytophthora, and Pythium species, during the first glasshouse trial to identify 5 trial sites with significant ARD for the next glasshouse trial. We planted apple seedlings in the collected untreated orchard soil and pasteurized soil. The increase in seedling weight and length for each treatment was determined over a 3 to 4 month period. The trial sites selected are those that showed infections of all 3 ARD causative agents and had a growth improvement of higher than 200%, which were identified as Nooit Gedacht, Vastra Block G3 and Tandfontein. We also selected Paardekloof and Kromfontein for the trial because they showed a high growth improvement in the seedlings and they had Pratylenchus infections of the roots. Soil has been collected from the selected trial sites and a glasshouse trial has been established where seedlings will be grown in the soil collected from the control, mulch and the compost and mulch rows of each site. Once again, we will compare the growth in the orchard soil and pasteurized soil, and we will identify the presence of a range of ARD causative agents from the seedling roots. The glasshouse trial will be repeated and statistical analysis will be done to determine whether the application of organic amendments does suppress the ARD causative agents. Root samples will be collected from the orchards to determine if the amended soils have decreased ARD pathogen inoculum levels. We will also be analyzing the soil quality by testing different parameters to investigate the associations between the soil quality and ARD suppressiveness of the organic amended soils. The soil quality parameters we will be analyzing are listed here. If the long-term application of organic amendments can reduce ARD causative agents in orchard soils, more environmentally friendly options than fumigation might be effective for establishing new replanted orchards. These include the use of compost plus mulch and omyceticides, which will likely provide more consistent and effective suppression of ARD in replanted orchards due to a reduction in pathogen inoculum in the soil during the lifespan of the previous orchard. I would like to acknowledge the following people, and I am looking forward to working alongside them throughout the rest of the study. Thank you.